everyone, and welcome to today's webinar in the CERDP and ESTCP webinar series. My name is Jennifer Nyman. I am a Senior Engineer at Geosyntec Consultants in Oakland, California, and the coordinator of the webinar series on behalf of CERDP and ESTCP. I will be facilitating today's call. The webinar will consist of a brief overview of CERDP and ESTCP by Dr. Andrea Leeson, followed by a list of upcoming webinars in this series. After Andrea's opening remarks, we will transition to the technical portion of the webinar. Today's event features 3 presentations on environmental health criteria, fate, and transport of insensitive munitions. First, Dr. Mark Johnson of the U.S. Army Public Health Center will present on the development of environmental health criteria for insensitive munitions. Dr. Johnson's presentation will be followed by a brief question and answer session. Then, Katerina Dontsova of the University of Arizona in Tucson will provide a presentation on the dissolution of NTO, BNAN, and insensitive munition formulations and their fate in soils, followed by a second question and answer session. The final technical presentation will be delivered by Dr. Jim Field, also from the University of Arizona, who will present on the biodegradation of NTO to mineral products, followed by a presentation-specific question and answer session. We will conclude the webinar with an interactive question and answer session with all 3 speakers. Today's broadcast will be listen only. You may submit questions by using the chat box on the left-hand portion of your screen. You do not need to wait until the question and answer period to submit your questions. And in fact, we encourage you to submit questions in advance of that session. On this slide, we have provided a few suggestions in the event that you experience difficulties with the broadcast audio. Typically, any delay will be fixed if you refresh your screen or call into the conference line again. However, if you continue to have problems, please submit a comment using that chat box in the lower left-hand corner of your screen. With that, I would like to introduce Dr. Andrea Leeson, who is the Deputy Director of CERDP and ESTCP, as well as the Program Manager for Environmental Restoration. Andrea has been with CERDP and ESTCP since 2001. Previously, she was a research scientist at Battelle Memorial Institute, where she conducted research on in situ bioremediation. Andrea?
Thanks, Jennifer, and welcome everyone to today's webinar. CERDP is the Strategic Environmental Research and Development Program, and we were established in 1991 by Congress as a partnership between the DoD, the Department of Energy, and the EPA. And CERDP's mission is to identify and address high-priority environmental science and technology opportunities that focus on our DoD requirements. We fund both fundamental research as well as advanced technology development under CERDP that ultimately impacts real-world environmental management. ESTCP is the Environmental Security Technology Certification Program, and in here we demonstrate innovative environmental and energy technologies. And our investments under ESTCP capitalize on past investments under CERDP or other research programs and are designed to transition technologies out of the lab and into the field. Especially important in all of our ESTCP demonstrations is the ultimate transition to implementation and regulatory acceptance. CERDP and ESTCP are complementary programs, with much of CERDP research occurring at the lab and pilot scale with occasional field efforts, while ESTCP demonstrations are primarily at the pilot and field scale, although occasionally we have supporting lab efforts. There are 4 program areas in CERDP, 5 in ESTCP. The Energy and Water Program area is only in ESTCP, while the other 4, Environmental Restoration, Munitions Response, Resource Conservation and Climate Change, and Weapons System and Platforms, are CERDP and ESTCP programs that are managed jointly by one program manager. Our webinar today is focused on research and demonstrations that were conducted under the Environmental Restoration Program area. And under environmental restoration, we have essentially 5 main areas of research, including contaminated groundwater, contaminants on DoD testing and training ranges, contaminated sediments, wastewater treatment, and risk assessment. Our webinar series will actually be highlighting research and demonstration efforts from each of the 5 different program areas, and shown here is a list of some of the upcoming webinars, which you can see cover a broad range of topics, with upcoming topics covering insensitive munitions, to geophysics, to remote methods for water conservation. The next webinar, based on research and demonstrations under environmental restoration, will focus on our Geophysics 101 webinars, and this will be a 2-part webinar on geophysics. Information about upcoming webinars on our CERDP and ESTCP website, and registration is now live for the webinars through August. I hope you enjoy the webinar today. Jennifer?
Thanks, Andrea. Thank you. It is now my pleasure to introduce Dr. Mark Johnson. Dr. Johnson is the Director of Toxicology at the U.S. Army Public Health Center, Aberdeen Proving Ground, Maryland. He has worked extensively on evaluating the toxicity of military unique compounds and developing and evaluating sensitive indicators of stress for use in field applications and toxicity testing. He has broad experience in risk assessment and has developed and tested new methods on improving exposure-effect relationships. Dr. Johnson has authored over 100 peer-reviewed publications, book chapters, and technical reports, and he is the current president of the American Board of Toxicology. Mark?
Hi, Jennifer. Thanks, and welcome, everyone. First, I'd like to begin with a little background on insensitive munitions. Insensitive munitions is an important area of research devoted to finding materials that only explode as intended for new weapon systems and platforms. And that's designed to reduce the very real and acute effects of munitions storages and storagings going up when bad things happen, whether it's because of accidents, because of terrorist incidents, or what have you. And so a lot of new compounds and new formulations are being developed to replace some of our current inventory to get these munitions and designed the way to only explode as intended. And so if you can see on that very first slide, the 2 formulations I'm going to talk about is IMX 101 and 104, and mostly IMX 101. 101 is really 3 components that we'll talk about in a few minutes. And what you see is actually the melt-pour operation, where these compounds are melted and poured into munitions. They're cooled to reduce that sort of peanut butter brittle-like flake material on the bottom right, and that then is transported to other facilities where it's melt and poured in specific munitions. And this particular work unit that we have been assisting on is to fill in some of the toxicity data gaps that exist for these compounds, because as there's a really immediate need for these materials, we want to all find a safe and sustainable way to use these materials from an environmental perspective. Slide 16, please. And so as I mentioned, IMX 101 is predominantly nitroguanidine, 2,4-dinitroanisole, which I'll refer to as DNAN, and 3-nitro-1,2,4-triazole 5-one, or nitrotriazolone, I'll refer to as NTO. RDX is substituted for nitroguanidine in the 104 composition that is typically only used as a primer for the 104 primary explosive. Immediately, as we were trying to transition this technology as quick as we could to the field, we realized we had some issues with occupational health. We didn't have industrial hygiene methods at some of our facilities, and therefore we also needed toxicity data to develop occupational exposure levels. And so that was one thing that happened right off the bat. But also, as we began to look at these compounds, we realized that 2 of them are very high in terms of their water solubility, that we had some toxicity data gaps that were incomplete, and that since these are insensitive munitions, the explosive ordnance disposal individuals who typically blow in place munitions that do not blow off as intended on our ranges, typically blow in place, requires a whole lot more C4 to get these munitions to go off and be rendered harmless, to the point where it just generates lots of dust. I mean, these things are truly insensitive. They only go off as intended, and so it becomes more of an exposure issue for these personnel who are involved in taking care of these munitions that don't go off. And so the whole goal was to help develop safe levels of exposure to fill in these toxicity data gaps and to enable developers to better estimate full life cycle costs. Next slide, please. So slide 17 shows some brief information on those three components I spoke of earlier. DNAN is the most toxic of the three from an acute standpoint. It's LD50, where median lethal dose is about 200 milligrams per kilogram in rodents. It looks very similar to traditional nitroaromatic compounds that we have been using, TNT, DNT. It's very similar in structure with just some slight differences. Its water solubility isn't too high. Nitrotriazolone, however, did not produce toxicity in any of the rodent studies that were conducted, going as high as 2,000 milligrams per kilogram. However, in the oral 90-day subchronic study, there was evidence that sperm production was halted at very high concentrations, above 300 milligrams per kilogram per day from 90-day exposures. Its water solubility, however, is very high, between 12 and 17 grams per liter. For nitroguanidine, I think it's fair to say it's fairly non-toxic. It did not produce toxicity in the acute studies, in the 14-day oral studies, even in the longer-term 90-day studies, near 1-year studies. Exposures up to 1,000 milligrams per kilogram per day did not produce toxicity. However, its water solubility is relatively high as well. Slide 18, please. And so our technical approach was to fill in a couple of these data gaps. Predominantly with NTO, we were very much concerned with reproductive and developmental effects, since it did show in a 90-day subchronic study that sperm was affected. And so we wanted to do this through the extended one-generation design using rats to apply to mammals and apply to humans. To do it also in a extended one generation in birds, so we had some criteria for birds, since birds are physiologically a lot different than mammals. We wanted to test it with them as well to see if that same mechanism was conserved in terms of reducing sperm counts. And we also looked at developmental toxicity to frogs, because if this compound was or worked through an endocrine disruptive mechanism, maybe potentially through the thyroid, you'd see it in frogs, because frogs are very sensitive to that. We also did not have any aquatic toxicity data for DNAN, so we also wanted to do that testing. Next slide, please. And so investigators at the Engineering Research Development Center of Vicksburg, Mississippi, Alan Kennedy and his colleagues, did acute and chronic testing in fathead minnows and Cereodaphnia. Basically, acute and chronic were fairly similar in the fathead minnow, which was very interesting. I'm sorry, but the Cereodaphnia, excuse me, acute and chronic were very similar, but fathead, there was a slight difference in chronic exposures. Slide 20, please. And as far as sublethal effects, we saw a reduction in growth in fathead minnows at around 12 milligrams per liter or greater. There was no survival impact to Cereodaphnia at longer-term exposures, and reproduction was just affected at around the same concentration, 2 milligrams per liter, which makes it a little bit less toxic than some of the conventionally used nitroaromatic compounds that I spoke about earlier. Next slide, 21. Here is the data I was describing about reduction in sperm counts and reduction. You can first initially see that through reduction in organ mass. And so if you look at the testes mass straight away, you can see a significant difference in rodents that were exposed to 350 milligrams per kilogram per day for 90 days and higher. You also saw a difference if you compared it to body weight at the same level. Also, if you want to compare it with brain weight, because brain weight is relatively consistent between animals with differing body sizes. And epididimides, you can see that where effects do occur somewhere between 100 and 315 milligrams per kilogram per day. Slide 22 shows histology of the testes. And if you look in the lumen, the center of these donut-shaped objects, you can see in the control animals, these little filamentous structures was actually the tails of the spermatids in the control animals. But in the very high dose, and I just showed the very high dose here so it would be easy to see, those spermatids just do not exist. They're not there. However, interestingly enough, in our reproductive developmental screen, we did pups born. And we attribute that to the fact that rats are just very, very good at reproduction. Even with very few spermatids, it doesn't take many to produce a full litter. Next slide, please. All right, on slide 23, what you see are the results of our endocrine disruption assays. This is what the EPA recommends as a Tier 1 screen. Most of these are in vitro assays. However, we did also do 3 in vivo assays in rodents, Hirschberger's, eutrophic, and prepubertal, all of which were negative. So this information suggests that NTO is not working through an endocrine disruptive mechanism. Next slide, please. And so in this study, we looked at conducting extended one-gen reproductive assay tests where males were exposed for about a month before exposure. Females a little bit less than that. We saw, by the way, no effects in the females whatsoever from exposure to NTO. But this study allows us to look at a lot of different endpoints simultaneously and to include a recovery group and some immunotox endpoints as well. Next slide, please. And so this slide here shows some of the predominant endpoints that we were focusing on in terms of reproduction. And I won't read every one of them, but a lot of data was collected in this study, and we don't have the time to go through all of it. And so if we go to our next slide, 26, basically it looks like there's a slight difference in survival, but statistically really not. These were drinking water studies, and in the high dose, animals just didn't drink very much, and so there was a little bit of difference, but not statistically so. No difference in pup mass whatsoever. But we did see changes in the F1 generation, that were the offspring, in terms of how long they retained their nipples and some prepubertal delays. Next slide, please. Okay, 27 shows the results of our avian toxicity extended one-gen, or actually the study design, which very closely mimics what we did in rodents. And we also added some behavioral assessment endpoints as well. Next slide. And the take-home from this is we saw something we did not expect. We saw neurotoxicological signs in our quail, where we saw some head tilt, some mild convulsion, seizure in the high-dose animals. In fact, none of the high-dose animals made it past week 5 of exposure. These were drinking water studies as well. No, excuse me, these were gavage studies. Only one male made it to the end of the study in the 500 milligram per kilogram per day dose group. And there we saw in that one individual changes in testes mass. But the fact that mortality occurs here makes the fact that the testes mass was changed in that one individual really inconsequential. But the fact that we saw neurotoxicological signs in the birds but never saw anything like that in the mammals was of interest to us. But keep in mind, this is still at a very high concentration. Next slide, please. Looking at amphibian toxicity, Dave Pillard at TRE Environmental conducted some studies in concert with us looking at the effects of frogs because, as I mentioned earlier, frogs are a very sensitive model for looking at effects through thyroid inhibition. And so basically tadpoles were exposed throughout metamorphosis to adult stage, and many different endpoints were looked at. Briefly, the results of this study are still being evaluated. In fact, the final report was just recently released, and no significant effects were seen up to 338 milligrams per liter. The main effect from NTO is that NTO does liberate a proton when put in water, and we think that's also was the reason why maybe it was a little unpalatable to the rats in the rat study. So it is a bit acidic. And the effects that we saw in the frogs based on the acidity was much more pronounced than any effect we saw from NTO by itself when it was buffered. And so most of the toxicity from NTO, at least in the aquatic format, is probably due to the fact that it is acidic when it's placed in water. Next slide, please. And so slide 30 are our basic conclusions, is that NTO with very high water solubility does create some fate and transport concerns, which our next speakers will speak to. And so we have some concerns about what's its probability, what's its environmental sustainability in terms of breakdown in the environment, how long will it bind to organic carbon, will it make it to groundwater? That's still a question. We did see that high doses, very high doses, cause a reduction in sperm production, and it seems to affect the Sertoli cells, which are the cells that produce and nourish sperm directly. And the high doses also were neurotoxic to birds. And DNAN in terms of aquatic toxicity was very similar or less than other nitroaromatics. Next slide, please. So slide 31. We've learned a lot since this. We've learned that it's really important for us to work with those in research, development, test, and engineering up front to use a phased approach to provide environmental safety, occupational health data in a very comprehensive but yet feasible manner, where in silico approaches, computer models can use to help out develop what kind of toxicity, what kind of environmental effects, what kind of occupational effects we could see from new compounds, even as early as this conception stage when molecules are just being developed on a computer. There are models that one can use now, quantitative structure-activity relationships and other models, to begin to provide this kind of iterative approach with this information to the developers and research to give them some information on whether or not this compound would be good from that perspective, along with their criteria in terms of efficacy and how it's going to work for their purpose. In the synthesis stage, we recommend that we do this to this ASTM 2552, which is a guideline that describes what I'm talking about, to begin to use some high throughput screening, maybe some in vitro methods, to begin to help in providing information in a relative way, what compounds look better from an environmental safety, occupational health perspective. And then the production and use stage where scaling up is feasible and it's worthwhile and it has passed most of the studies that the principal investigator needs it to pass for its intended purpose. And at that point, maybe some very focused in vivo work is then needed to be able to provide the kind of information we need to protect the workers and protect the environment. And I believe, with the next slide, please. And here's the additional information at the website. As we finish our reports, we'll try to put them there for you. My contact information is below, and if you have any questions, I'm happy to entertain them. Thank you.
Thank you, Mark. We have received a few questions, and I would also like to remind our audience to please submit questions by using the chat box on the left-hand portion of the screen. So Mark, the first question for you is, have any environmental criteria for these constituents been developed?
Well, no criteria have yet been developed by any regulatory agencies. However, our agency has developed a couple preliminary guidelines for purposes if there are environmental releases at some of our munition plants. And basically, there were risk-based concentrations for industrial scenarios. There's also, I think we also calculate those for residential scenarios, but that has not been an issue yet. But basically, soil concentrations and surface water concentrations. But that, to my knowledge, is all that's been developed so far.
Okay, thank you. The next question is, do mammals recover when exposure ceases?
Okay, we did have a recovery group in the extended 1-gen that suggests that possibly. We saw some damage to Sertoli cells, and the available literature suggests that if exposure continues for an extended period of time, you could have permanent damage to Sertoli cells where the cells don't die, they just do not function. And so no sperm is produced after a long period of time exposure. What that period of time is, we don't know yet. If it's a short period of time, yes. It is likely that there's recovery.
Okay. And what is the likelihood that environmental releases would be toxic to wildlife?
Okay, if I were to speculate, I would say not likely at all. The concentrations that would be required to see adverse effects to wildlife would be incredibly high for sustained periods, particularly for compounds like NTO, nitroguanidine not at all, and DNAM probably less so because of its environmental fate and transport, which our next speakers will address. But I don't think it's very likely at all.
Okay. And Mark, can you comment on what are the next steps for these evaluations for these specific insensitive munitions or for this topic more broadly?
Yeah, great question. Well, for us, of course, we're concerned about environmental exposures to humans as well as other organisms, but we think it's important to have some chronic data. We have subchronic data. We have extended 1-generation data, but at this point, I think we need chronic data to be able to develop criteria that would be appropriate for the general population. Typically, that's what the EPA likes to have before they develop reference doses or drinking water health advisories and that sort. And so it's in the plans for us to do that with assistance from the Army Environmental Command. We hope to do that this fiscal year, begin that. But it's going to be a long study. It's going to take us a couple years to get that one done.
Okay, thank you. And following up on your presentation, we have a more specific technical question about NTO. Which is more important to aquatic life? Is it the toxicity of NTO or the acidity of it?
I think it's clear that the acidity is more important than the buffered toxicity of NTO. I think that's fairly clear. And I guess the questioners was in referencing aquatic toxicity. Yeah, and there was a study conducted by Mark Haley at the Edgewood Chemical Biological Center, and that report is out, where he also buffered it and saw at least an order of magnitude reduction in toxicity once it was buffered.
Thank you, Mark. And Mark will be back at the end of our seminar to answer additional questions. And thanks, Mark, for the presentation and answering these questions.
You're welcome.
The next part of the webinar will be presented by Dr. Katerina Dontsova, who is an Associate Research Professor at the University of Arizona in Tucson. Dr. Dontsova's research focuses on insensitive munitions. Her previous work examined the fate and transport of energetic compounds in soils, including traditional explosives and propellants. She employs numerical modeling to quantitatively characterize dissolution, adsorption, and transformation of these compounds. Dr. Dontsova has authored and co-authored multiple publications that examine the fate of munitions in soils. She graduated from the Ukrainian Agricultural University in 1995 with a degree in soil science. And she completed her master's and doctoral degrees in soil chemistry at Purdue University. Katerina?
Thank you. Good morning. I am presenting results of SORTA project AR2220. In this project, we were interested in release and natural attenuation of these new insensitive munitions in the environment. To determine their impact of any contaminant on the ground and surface waters, we need to know what is the source of this contaminant and then how much of the contaminant will be naturally retained or transformed in soils, preventing its impacts downflow. Since the common source of energetics on the training ranges is the deposition of the unexploded fill during incomplete detonations, what we did is first examined the insensitive munition dissolution under controlled conditions. We looked at outdoor dissolution phototransformation. For the NTO and DNAN, we did soil absorption studies as well as transport studies, and then we looked at the dissolution and soil transformed of IM formulations. We can predict something about the environmental behavior of these compounds just from their structure and their physical properties. For example, we know that insensitive energetics have high solubility, particularly for NTO, as Mark mentioned. So fast dissolution and high mobility in soils might be expected. The second, for the DNAN structures here, it's a natural aromatic compound, similar to TNT and DNTs, the traditional energetics, and we can expect that it would have similar behaviors in soils. We know that TNT tends to be retained and transformed in soils, and it is not very mobile. NTO, on the other hand, can deprotonate. It can lose proton and nitrogen there, and then it will become negatively charged. Soils are also negatively charged, so we would not expect them, NTO, to be retained in the soils. Deposition of NTO and DNAN on training grounds is, as I mentioned, is expected in solid form in IM formulations. So we need to know what is the structure of these formulations and their composition. 3 formulations have been developed. Here are IMX 101, 104, and PAX 21. PAX 21 has been since discontinued due to potential for mental contamination. All 3 of these IM formulations are melt cast, or Mark called it melt and pour, which is the same thing, similarly to composition B. So in this process, DNAN serves as the matrix, and in this matrix, NTO and nitroguanidine in case of IMX 101, NTO and RDX in the case of 104, ammonium perchlorate and RDX in the case of PAX 21 are dispersed. So there are significant differences compared to traditional munitions. One is that most IM components, except for RDX here, have higher solubilities than traditional munitions. And second, that component with smaller solubility, DNAN, serves as a matrix. So that will slow down the dissolution of other components. Logically, when IM formulations are exposed in the environment, they are dissolved in natural precipitation, they are phototransformed in solid and solution form, and then they interact with soil. So we designed our studies to examine these processes. To determine mechanisms of ion dissolution, Dr. Susan Taylor in Cold Region's lab performed lab dissolution studies that looked at how ions dissolve and how the structure of these particles changes as a result. So here on the left is the X-ray microtomography images of the particles of three formulations, 101, 104, and PAX21. On the top is the amount of water that they've been exposed to. The particles are about 2 millimeters in diameter. So you can, on the images, you can see these brighter particles. So these are the crystals of NTO and NQ in IMX101. It's NTO in IMX104 and ammonium perchlorate in PAX21. And so they dissolve first. This is particularly clear in the case of the PAX21, but also visible for other particles. And so this leaves behind these pores, empty spaces within the particles. Also visible on these plots, you can see the cracks that are running through the particles, which are a result of the detonation. So that would allow water to get inside the particle. On the right is the amount that was dissolved from these particles, and it's the cumulative dissolution in milligrams versus water volume. And you can see again the same pattern, so NTO and NQ dissolve fast here, and ammonium perchlorate here, and then it's followed by DNAN or DNAN and RDX in the two other compounds. So what we would expect that for NTO, we would see high concentration for a short period of time initially, but for DNAN, we'll see prolonged release and lower concentrations overall. During outdoor dissolution studies, when particles were exposed to rainfall and snow and sun, the similar trends in dissolution were observed with more soluble components like NTO dissolving first, leaving RDX and DNAN behind. About 65% of particle mass was dissolved in a little more than 2 years. That is about 10 times more than was observed for traditional explosives, which was about 5% in 3 years. So here shown is the IMAX101 particle, how it changes over time as exposed to the environment. So you can see that it's crumbling as time goes by. You can also see that there is a change in color. So this indicates phototransformation. Insensitive munitions, on average, lost about 15% of initial mass to the phototransformation. That is equivalent to 19% of total mass that was lost from the particle, and a lot less than 74% for traditional explosives, which because of the faster dissolution of ion munitions. We identified some products of transformation, such as isomers of methoxynitroaniline, also called aminonitroanisole, methoxynitrophenol, and more rarely dinitrophenol. As Mark mentioned, NTO dissolution has been shown to impact pH of solutions. So if you look here, this is the result of both lab and outdoor dissolution studies. And you see a relationship between pH and NTO concentration. As NTO concentration increases, the pH goes down to quite low values, presenting an environmental problem, but it also gives an opportunity for potentially to develop a field method for measuring NTO just based on pH of solution. In the soils, we did not see significant effect on pH, and this is likely due to the buffering of solution acidity there. So potentially the larger effect of this acidity was going to be on the quality of the surface water rather than for the groundwater. These compounds can phototransform also in solution phase, because before the studies that I showed looked at the solid phase phototransformation. So when there are solutions, they can also phototransform, and their products can be either complete mineralization or it can be formation of organic daughter compounds. Dr. Persia Rodriguez observed that there was a effect of pH and organic matter content on phototransformation of NTO, shown here. So at extreme pHs, there was more phototransformation, and as well if there was a presence of the humic acid. Neither of these had effect on DNAN, but for DNAN, as you can see here, the half-life were changing with temperature. For the NTO, you see the half-life is 5.3 days. over the range between 25 and 70 degrees. But if you look for the NTO, this line here is that there is a decrease in half-life with increase in temperature. So that would indicate that the persistence of D9 would be much lower in hot climates. And in fact, we're having some experiments done right now looking at these effects. Once dissolved, potential for natural attenuation of energetics in soils depends on soil properties, since they're very variable. We're very grateful to Army National Guard Environmental Directorate for collecting soils across the United States on National Guard sites that are shown here on these maps. So this allowed us to first determine potential attenuation of ions in these soils, but also it allowed us to develop relationships between soil properties and the fate of ions in these soils. So this plot on the left for absorption coefficients for NTO, so this is a measure of how much of this compound will be in the soil for a given solution concentration. It shows that NTO is very weakly absorbed, so these values are less than 1. And we also see some relationship with the pH of the solution. So if we go to higher pHs, we have less absorption happening. And this has to do with the change in the protonation or charge of NTO with soil pH. For D9, and these are the two plots on the right here, we see that the values are greater for absorption coefficients than for NTO, and the values are similar to what you expect for the TNT. And there's a strong affinity with organic carbon as well with the clays in the soil. So there is increase in absorption when you have higher organic carbon soil. To see the soil interactions, how this is reflected in the transport of ion compounds, we wanted to conduct common studies. There's a simplified models where we look at the patterns and we also determine transport parameters that can be then applied in the field. For the NTO in two soils, and this is Kaplan soil from Midwest, it has very high organic carbon content. On the right is sassafras soil, low organic carbon content. This is from East Coast, Aberdeen Proving Ground. So what we plot here is the amount of water that went through. You express this for volume. You can also express this time. And on y-axis, we have concentration of the compound as relative concentration relative to the original input concentration. And what we would expect if we have absorption, so this blue line for the NTO is going to be shifted right to the gray line for the tracers that did not react with the soil. As you can see, there is very little shift. In both cases, a little bit more for sassafras soil, indicating that there is very little absorption, consistent with what we saw in previous studies that I showed. There is some transformation. So this, if the line is lower, it means more of the compound has been transformed. And we see even for one soil with high organic carbon content, we see complete attenuation of NTO. In soil, so on the top, on the bottom, we have the sterilized soils, the same soils as above. And we can see that the values for transformation are much lower. And that, of course, indicates that we have a contribution of microorganisms for transformation. The shape of the line here as well indicates this too. This is for application of Monod kinetics. The same plots for D9, we see a lot more absorption. It's expressed as a retardation. So the retardation factor is about 6, indicating that it will take 6 times more time for D9 to arrive to the groundwater at the bottom of the column than it would be for tracer. And this was for the Camp Swift soils, which had very, very little organic carbon content, about 0.3%. So this is a very conservative estimate. We would expect that for soils with high organic carbon, as was shown before, would have a lot more absorption, so the retardation is going to be much higher. We also observe this in here in gray on both plots. We see a reduction of the nitro groups to amino groups with the formation of aminonitro anisole. to look at the patterns of contamination that can result of the combination of dissolution and soil detonation, we put a 50 milligram particle of MX101 here, no, MX104 here, on surface of the soil column, and then applied solution, or just rainwater. And we see very high peak initially for the NTO. This is at almost 2.5 grams per liter, which is followed by more steady state concentrations later on. So the mechanism behind this pattern is that when the outside crystals of the NTO are dissolved, then in order for the NTO to get to the edge of the particle, it has to diffuse through it. And so that takes some time and slows down the dissolution. Similar pattern is observed for NQ here in red. You would notice that the concentration of NQ is lower than for NTO. But as Mark mentioned, there's a lot more of NQ than NTO in IMX101 particles. So this lower concentration has to do with lower solubility of nitroguanidine. For D9 in green in both of these plots, you can see a pattern is quite different. So we don't see any of the original peaks. So the lines are fairly steady. So we have fairly low and fairly constant concentrations coming on for both of them. And you can see that there is a delay here for when they appear. The same is observed for RDX in orange here. Concentrations are even lower. And on both of these, we see formation of this product that I mentioned before, the amino nitroanisole, but the concentrations are fairly low relative to the concentration in the of the parent compound. So to conclude, based on this study, we found that IM constituents dissolve sequentially as predicted by their solubility. There's the fast removal of most soluble components first, and that creates a porous structure that can break up, and then that would further promote dissolution. The phototransformation of outdoor insensitive munitions was evident, but its contribution is smaller than for traditional explosives because we see faster dissolution here. Thinking about absorption for the NTO, it was very, very low, so most negligible, but it did depend on soil pH. For the DNAN, there was a greater absorption, and it was quite strongly related to the amount of the organic carbon or clay in soil, both in fact. Both of them were transformed in soil, and we see products of DNAN transformation in solution. So based on all of this, we have high release of NTO and low attenuation in soils, particularly if they have low organic carbon, as sediments would. And so that creates a higher risk of its transport to the ground and surface waters. While for DNAN, there is a slower dissolution and TNT-like behavior in soils. So that indicates that we'll have lower potential for off-site transport and greater natural attenuation. I would like to thank my collaborators and students who worked on the project. And if you have any additional questions about the studies, you can find information on the sort of website. The link is provided here. And thank you very much.
Thank you, Katerina. And I would like to remind our audience at this time to please submit questions using the chat box on the left-hand side of your screen. And Katerina, we have received some questions from the audience. They are specific to results shown on certain slides, and the first is on this slide, slide 41. And Katerina, the question is, since NTO is an anion, would it be expected that NTO dissolution would be more pronounced at higher pH?
I do not think we looked at the effect of pH on dissolution. So I don't know.
Okay, so this is the resulting pH as a result of dissolution?
Yes. It's not well, the pH of the water is going to be rainwater, which is going to be 5.6 at equilibrium with the CO2. So it's slightly acidic, around somewhere in the middle of the plot here. And the pH then is driven down by NTO itself.
Got it, thank you. Okay, and the next question is on the following slide, slide 42. Katerina, do you have any thoughts regarding why dissolved NTO would photodegrade fastest at the highest and lowest pH and more slowly at pHs in between that?
I'm not a specialist in phototransformation. I believe this is a requirement for things that can deprotonate to study them at different pHs, that it's expected that they would respond differently.
Okay, thank you. The next question is, how does the size of the particles studied in your work compare to what can be observed as a result of incomplete detonation in the field?
Yeah, the particles that Dr. Susan Taylor looked at and presented here, so this was for smaller particles it was about 2 millimeters, and for the larger it was about 2 centimeters. So this is about the range of the particles that would be deposited. It really is a distribution, so you would have from very small to larger particles, and 2 centimeters is probably on the upper range of what you would find. And the most of the number of the particles is going to be in the smaller sizes, but these larger particles have larger mass, then the contribution to the overall mass would be still significant for larger particles.
Thank you. Can relationships for soil absorption developed in this study be used to predict NTO and DNAN behavior in other soils?
Yes, if we look at the slide where I show this information, just give me a second to locate it, here. So if you look at this slide, you'll find that there is a very strong linear relationship between absorption coefficient and organic carbon. The soil that we were collecting was from all over the United States, so the range of the properties is very wide, which means that these soils are fairly representative of what you will find in different locations. And the variability of the measured value versus the lines, predicted lines that we did, is maybe the maximum is twice. So I think that means that we'll have a very good predictive capability for the absorption over a range of soils for different locations.
Thank you. And following up on that, do some of these results have implications for the remediation of these constituents in soil? So for example, in situ remediation of NTO or DNAN.
Do you mean soil absorption?
Correct.
What would
any
have effect?
Any kind of soil treatment of the constituents.
So, I mean, if it's absorbed in the soil, it would have a longer residence time in the soil overall. And so potentially as it dissolves, it can be remediated. I think probably it's harder to remediate if it just flushed through the soil and doesn't have a lot of chance to interact with whatever methods they use for remediation.
Thank you. And a question about the transformation of NTO and DNAN. Can we predict the rates of NTO and DNAN transformation in other soils besides the ones discussed in this presentation?
So this is a lot harder than with soil absorption. We did some relationship. We see some relationship between the transformation rate and the properties of the soil. Particularly for DNAN, we did see that it was a greater transformation when with transformation rates with high organic carbon. I'm not presenting here because the relationship is weaker than we see for the soil absorption. We also saw that when we have a lot of organic matter in the soil, for the DNAN transformation, there was also some faster change, which indicated there's a greater bacterial activity that's happening, and you have more transformation of the NTO. But that was fairly condition specific, and so the rates, while there were relation between the rates with condition, with the amount of the organic carbon, that was also dependent on how we did the experiment. So I think it's harder to predict the transformation rate than it is soil absorption.
Thank you. Is it possible to estimate total release of insensitive munitions from dissolution information and the total mass that was initially deposited?
So this is not very straightforward, but probably possible, because there is information, not from this study, but from a different study, which looked at the distribution of the deposition of these particles, we know that there is a relationship between the amount of water that was applied and D9 dissolution, since D9 has a fairly steady state dissolution like NTO. So we can relate amount of water, so like rainfall, which you should know. And so knowing the particle size distribution and knowing how much of D9 dissolves versus the amount of water applied, we can probably make an estimate on how much is going to be released.
Thank you. One final question for you, Katerina. What are the next steps for this work on looking at the dissolution and fate of these constituents?
So we're doing a study right now. The study for the dissolution that I showed you was done in New Hampshire, which is fairly high rainfall and fairly cold. So part of the time it's covered in snow. So probably little is happening. So what we're looking at is what would happen if you go to warmer climates. We'll have maybe less dissolution because we'll have, well, less rain, but we'll also have higher temperatures, so that and more sun. So high temperatures, as we saw, promote phototransformation of D9. More sun would affect also the phototransformation. So we might see a greater contribution to phototransformation relative to the dissolution of these components.
Thank you, Katerina, for answering the questions and for the great presentation that you gave. Katerina will return at the end of the webinar to answer additional questions. So if you do have more questions for her, feel free to submit those in the chat box, and she will be back later. Our final presentation today is by Dr. Jim Field. He is a professor at the Department of Chemical and Environmental Engineering at the University of Arizona. Dr. Field conducts research in the areas of bioremediation, biotransformation, and biodegradation of hazardous environmental contaminants. Among his current research topics is the environmental fate of the insensitive munitions NTO and DNAN. Dr. Field has published 245 peer-reviewed journal publications, including on the biotransformation and biodegradation of DNAN and NTO. He obtained his bachelor's and master's degrees from Virginia Tech and his doctoral degree in the Netherlands. Jim?
Thank you very much. I'd like to get started with an overview of our startup project. What we study in this project is the fate of IMC compounds in soil. One of the things that we're interested in is how these compounds get converted to other compounds, so how they become biotransformed. And an interesting question is whether they're still toxic when they do become biotransformed. The other issue that we're studying is how such compounds become reversibly adsorbed to soil particles. But also, we also observe that some of these intermediates become irreversibly covalently bound with soil humus, and we refer to that as bound residue. What we'd like the most is that the compounds just become completely biodegraded to mineral products such as CO2 and ammonium, shown here. Now, because of the time allotment, I'm just going to focus today on NTO because we have less information about that biodegradation of that kind of compound compared to DNAN. So here you see my objectives for today's talk. We're interested in establishing if it's possible to have biodegradation and biotransformation of NTO in soil. One of the guiding principles of our research group is that if we're able to reduce the nitro group of explosive compounds, then we can make the molecule more prone to oxidation. So therefore, we're interested in the sequence of anaerobic, aerobic pathways for the complete biodegradation of NTO. If we're able to demonstrate reliable biodegradation, then the next question is whether it's possible to achieve this in a bioreactor. And lastly, we're interested in knowing what microorganisms are involved in the observed biodegradation. In this slide, you see our methodological approach. The big working horse in our lab are these little serum bottles where we develop soil suspension microcosms. and we control the headspace so that we can study biodegradation and biotransformation either under anaerobic conditions or aerobic conditions. The main measurement techniques that we're using to measure the NTO as well as its daughter product is HPLC diode array detector. We use a hypercarb column for that. And some of the other methods that we utilize that we'll be discussing today is the measurement of CO2 with a gas chromatograph, thermal conductivity detector, and inorganic ionic forms of nitrogen, ammonium, nitrite, and to measure that we use the ion chromatograph. This is my first results slide, and what I'm trying to show you in this slide is that it is possible to influence the biotransformation of NTO in soil by simply adding an electron donating substrate to promote the reduction of NTO. So in this particular example, we've added hydrogen gas as the electron donating substrate, and the microorganisms, after a lag phase of about 3 days, rapidly reduce NTO, and they form one product almost completely stoichiometrically, and that product is the same molecule as NTO, except now the nitro group has been replaced with an amino group, and that product we abbreviate as ATO. And you can see that as the NTO is disappearing, it's being replaced completely by ATO, which is the final product. Now what happens if we didn't add any electron donating substrate? Then you see that there's a slow conversion of NTO in the right-hand panel, and it too is slowly being converted to ATO. In this particular example, it is a soil that has a high organic carbon content. If it were a soil with a low organic carbon content, then there'd be no conversion at all, period. All right, so alluding to what I mentioned previously in the introduction, the importance of having a proper redox sequencing of events, we're going to test the idea of sequencing anaerobic, aerobic stages of biodegradation to promote the full biodegradation of NTO. So in this experiment, we provided electron donating substrate. In this particular case, it was pyruvate. And we see that with pyruvate and the NTO was rapidly transformed, being converted almost stoichiometrically to ATO. When that process was complete, then we switched the conditions of the culture and made it all of a sudden aerobic, and then we see that the ATO then disappears, such that by day 37 it's all gone. Now, how do we know that this was important? Well, we know it because if we didn't do the proper sequencing, then we don't get the full biodegradation. So on the left-hand panel of this slide, you can see what happens if we only provide the anaerobic conditions. Then in the beginning, we do have NTO reduction. It is converted to ATO. However, once the ATO is completely formed, then nothing happens after that. It just sits there, and such that we just have ATO at the end of the experiment at day 37. What happens if we just provided aerobic conditions only? Well, in that case, we have no conversion. So we just have NTO, 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 such that by day 37, we still have all the NTO that we had at the very beginning of the experiment. So now that we've established that with a sequence of anaerobic, aerobic conditions, it's possible to have biodegradation, now the next question is it possible now to achieve this biodegradation in a bioreactor? So in order to study this, we've decided to work with a biotrickle reactor that has these 1 centimeter squared, 1 centimeter cubed foam blocks. We put the IMC compounds in this influent container, and we put it into the reactor with a shower head, and it's trickling through these blocks, and it also gets recirculated. There's also a counterflow of humid air coming in from the bottom and going up. Another thing I wanted to mention is that the empty bed hydraulic retention time in this bioreactor is 42 hours. So in the beginning, we started with ATO as our only substrate in this reactor, and the logic here is that we're trying to build up a population of healthy aerobic ATO biodegrading microorganisms. So over the course of the first approximately 200 days, we're just feeding it with ATO, and the influent concentration is the blue diamonds that you see here, and the effluent concentration are the red squares that you see here at the base. So basically what you're witnessing is that over this whole period, we're getting approximately 100% removal of the ATO. Now, what we did next is we switched the IMC compound. So now, instead of adding ATO, we switched it, and now we're going to start adding NTO. Remember that you need electron donating substrate, so we also added pyruvate to provide the electrons for the reduction. So what we see here is that it takes a while for the NTO reducers to establish themselves, but we see that over a course of about 60 to 70 days, a population is coming in, and the NTO and the influent is shown in the red squares here. The NTO and the effluent is shown by these red X's. So we see that there's steadily more and more removal of NTO over time, such that by the time we get to day 300, we're getting complete removal of the NTO. Now the ATO and the effluent is shown by these blue X's right here on the bottom of the axis. We see no accumulation of ATO, so that means that the NTO is being converted to ATO and then subsequently fully biodegraded. In order to prove that there's extensive biodegradation taking place, we demonstrated nitrogen mineralization in the bioreactor. This graph is comparing the elimination of the IMC compounds with the release of mineral nitrogen compounds such as ammonium, nitrite, and nitrate. So the green squares is showing you the percent removal. So remember when we were adding ATO, it was 100% removal. Remember when we added NTO, initially it wasn't working so good, but then eventually it started catching on, such that by day 300 we had 100% removal. So now we can compare the percent removal with the percent release of inorganic nitrogen, and that is shown in the blue diamonds, and we can see that we're getting from 40 to 50% release of ionic inorganic nitrogen species. Now to prove the point even more, we developed enrichment cultures that were specialized in degrading ATO aerobically, and these cultures sustain themselves. We're on transfer 30 today, but a couple of weeks ago when we were on transfer 26, we did an experiment where we compared the ATO degradation with the release of CO2. So we see that we inoculate this enrichment culture on a transfer. We have a lag phase for the population to start to grow. Then the population gets big enough, and then all of a sudden ATO disappears. And just at the moment it's disappearing, we're seeing accumulation of CO2 in the headspace, such that we have 1.2% CO2 in the headspace. We can use Henry's law and the dissociation constant for H2CO3 to calculate the expected CO2 if 100% release of carbon occurred, and we calculated 1.2%. So this slide demonstrates conclusively that the organic carbon in ATO is converted completely to inorganic carbon in CO2. We did the same experiment in another transfer, but this time we're monitoring the release of nitrogen. So in this experiment we see that the ATO is being degraded. Why it's being degraded, we're getting a release of ionic inorganic nitrogen species, in this case mostly ammonium, such that by the end of the experiment we have a release of approximately 50% of the nitrogen. We also conducted a clone library of this enrichment culture to find out what kind of microorganisms are present. We identified 3 operational taxonomic units that had a greater than 99% sequence lineup with known bacteria. The first 2 of these were Hydrogenophaga and Ralstonia. And it just so happens that these bacteria are known as a co-culture in the literature to degrade this compound right here, which is 4-aminosulfonates. And so that makes these organisms very suspect of being involved. And the third operational taxonomic unit that we found was Nitrospira, which is a well-known bacterium, nitrifying bacteria that oxidizes nitrite to nitrate. So that brings us to the conclusions of our talk. We see that it's feasible to have full biodegradation of NTO with the proper sequence of anaerobic and aerobic phases. We've demonstrated the complete mineralization of ATO organic carbon to CO2. We've demonstrated extensive mineralization of the ATO nitrogen and subsequent nitrification of that nitrogen. Hydrogenophaga and Ralstonia are implicated as the suspects responsible for ATO degradation. And we also have demonstrated that it's possible to have NTO be converted to ATO and subsequently completely biodegraded in a biotrickle reactor. And later I'm going to show you a pictogram that explains how that's possible via anaerobic microniches. And the pathway that we've described through our study is shown here. In the anaerobic biotransformation phase, we have the conversion of NTO to ATO, just the reduction of this group to an amino group. The mineralization of ATO might have organisms such as Hydrogenophaga and Ralstonia implicated in the process, and Nitrospira implicated in the nitrification. I was asked to provide a slide for suggestions to practitioners. So my suggestions are as follows. If you want to promote the rapid biotransformation of NTO, you would need to add electron donating substrates in order to achieve that. After that reduction event, then you need to provide aerobic conditions in order to get the oxidation of the ATO. One of our future goals is to develop a primer set to detect ATO degrading bacteria at any given site. That's important because we've noticed that the ATO degraders are not present at all sites. Therefore, sometimes it might be a requirement to bio-augment ATO degraders at specific sites that are lacking the ATO degraders. We see that it is possible to have the full biodegradation of NTO in a bioreactor, but we must have both the aerobic and anaerobic zones in the reactor to reliably achieve this biodegradation. In pictograms, suggestions for the practitioners are as follows. So if we have a field site, then to get the bioreduction, we need to inject electron donor into the groundwater in order to achieve the reduction. Not very different from what's being done with chlorinated solvents. However, when that reduction is complete, then we have to provide aerobic conditions in order to stimulate the mineralization of the ATO molecule to CO2 and ammonium. In a bioreactor, it's important to provide both aerobic and anaerobic zones. With the foam block approach, we build up a biofilm on the foam blocks. We are providing pyruvate in this particular example as the electron donating substrate. So we have hundreds of milligrams of pyruvate, but just maybe maximally 8 milligrams per liter of oxygen. So obviously for, the diffusion of the electron donor is going to go much deeper than that of the oxygen. So we're going to have a zone here in the middle where we have an anaerobic zone. This is where we're going to be able to achieve the reduction of NTO to ATO. Then when it diffuses back out into the aerobic bulk phase, that's where we're going to get the mineralization of the ATO to CO2 and ammonium. I'd like to thank all of the project team partners that were involved in the research. They're listed here. And on this slide, you can see the link to our project, and you can learn more about our partners that are involved in the project. And also we have this lab website here on the bottom, which lists all of our publications, so you can find a lot of the information in some of these publications. And I believe with that, it concludes my talk, and I'll be happy to answer any of the questions you may have.
Thank you very much, Jim. And we have received a number of questions for Jim. The first is, what are your thoughts regarding the natural degradation of NTO in aerobic groundwater? Would there be very slow, or would there be any degradation?
Yeah, it sort of depends on how you define aerobic. So if it's aerobic in the sense that there's absolutely no co-substrates at all, then I do not expect in such an environment rapid NTO degradation. There you would need to enhance the process by adding electron donating substrates.
Okay, thank you. And what electron donating substrates are suitable for NTO biotransformation?
Okay, so when I showed you my first results slide, I had a reference there, and you can also find it in our website, Chris Marzak from 2015. If you go to the supplementary information in that article, you will find a large array of electron donating substrates that we tested. I think we tested more than a dozen substrates that are usually very good for chlorinated solvent dechlorination are typically also very good for NTO reduction. So, you know, the usual suspects, ethanol, lactose, lactate, butyrate. The ones that are bad are the ones that are also bad for chlorinated solvents, so just adding acetate is not very good. Methanol was not very good. So you can look in full details if you go to the supplementary information of the Chris Marzak paper from 2015.
Okay, thank you. If all the organic carbon is mineralized to carbon dioxide, what is the form of the nitrogen that is not mineralized to either ammonia or nitrate?
Okay, so that's a very good question. We don't have the complete answer yet, and the reason why we don't have the answer is because the way that we measure nitrogen and oxygen, they come out at the same time. So in order to answer that question, we either have to get another column that separates nitrogen and oxygen better, or I have some other ideas about how to take a sample and eliminate all the oxygen and then measure the remaining nitrogen. So we're still working on that, but the obvious suspect would be dinitrogen gas.
Okay, thank you. Was the NTO reduction to ATO co-metabolic, or was it energy yielding? And do you know if it was carried out by the ATO degraders or different organisms? Maybe two
we do have, so at first we thought that, wow, this could definitely be a reaction that's gaining energy. So we set out some experiments to test out. And so the way we checked out was by doing an enrichment culture under NTO reducing conditions, supplying an electron donor. And then in one enrichment culture we supplied the NTO and the other one we didn't. And we looked at the impact that had on the population, and the populations were exactly the same. It didn't matter that you added the NTO or not. So from that, we conclude that it's not an energy yielding process where an organism is benefiting from reducing the NTO. What we conclude is that we're just creating a population of anaerobic, an anaerobic consortium of microorganisms by adding the electron donating substrate that are responsible then for this co-metabolic reduction of the NTO. So that's the situation that's closest to the reality.
Okay, thank you. In a soil environment, would you expect significant absorption of ATO? And as a follow-on, would an important component of that be humification?
Okay, so that's an interesting question. So we have these two families of IMC compounds. One is the NTO family, and the other is the DNAN. So the business about the humification is very relevant for the DNAN family. However, for the NTO family, we don't expect humification. We expect mineralization of the compound. In terms of sorption, so when we talk about sorption, it's reversible sorption now that we're talking about. ATO doesn't absorb very well to anything in the soil. The only thing where there's a little bit of absorption is with iron oxides. And that matter, NTO, absorbs much better the iron oxides than ATO, but the ATO does have a little bit of absorption too. Not very powerful, but it absorbs a little.
Okay, thank you. And what enzymes convert NTO to ATO? Do you have any information on that?
We don't, but we would expect it to be some kind of reductase. Since it's co-metabolic, it doesn't even have to necessarily be a very specific enzyme. It could just be, like some reduced flavin group and some enzyme is just reacting with the nitro group.
Why are explosive molecules more prone to oxidation after reducing the nitro group?
The reason that's the case is because the nitro groups are sucking away all the electrons from the skeleton of the molecule. So if they're sucking away all the electrons from the skeleton of the molecule, then the skeleton of the molecule is actually more oxidized than normally without that nitro group. So when you have a compound that's already partially oxidized, it's much more difficult to oxidize than if it weren't. So if we were to reduce the nitro group and make it into an amino group, then we have an electron donating group, and so then we're pushing electrons into the ring, and we're making it much more prone to oxidation.
Okay, thank you. One more question from the audience. Are intermediates expected during mineralization of NTO?
We have, along the whole process from NTO to ATO and then ATO subsequently to CO2 and ammonium, we have found 2 instances of intermediates. So in the first step from NTO to ATO, we have found small amounts of the hydroxylamine. So that means that the nitro group is reduced by 4 electrons instead of 6. However, that's just a fleeting intermediate that later gets reduced again. Between ATO and the mineralized products, we have detected very small concentrations of another intermediate. We don't know the exact structure yet, but it contains nitrogen and sulfur.
Thank you. Under what circumstances does NTO occur as the main contaminant?
Well, given the previous talk, we see that if we have undetonated material and then it's getting dissolved in the rainfall, clearly the NTO is getting released first. So it's traveling out way beyond the other constituents, and then when it goes into the soil, then you kind of have like chromatography. So the NTO is going just shooting through really quickly because there's no retardation. And then the other constituents, such as DNAN, then dissolve slower in the first place, and then when they get to the soil, they're retained a lot by the chromatographic column of the soil, and so they retard a lot. So I would expect that you could have situations where the NTO goes way out and contaminates groundwater and surface water where there's no DNAN present.
How are anaerobic micronesia created, both in the natural environment as well as in the engineered systems for treatment?
So the main thing is if you have electron donating substrate, then you can create anaerobic micronesia because first of all, there's 2 things going on. One is that the substrates consume oxygen, so that gets rid of the oxygen. And then the other thing is if you have a biofilm in a bulk aerobic environment, the little bit of oxygen that's present will only be a few milligrams per liter. So that means the driving force for diffusion into the biofilm is not very strong. However, if you have an electron donating substrate, it will be present at hundreds of milligrams per liter, and that's a very good driving force for diffusion. So the electron donating substrate is going to diffuse deeper into the biofilm than oxygen, and that's how you create a microniche, an anaerobic microniche.
Okay, thank you, Jim, and thanks for answering questions about your presentation. At this time, we'd like to bring back Katarina and Mark and pose some general questions for all 3 speakers. The first question is going to follow directly on Jim's presentation. The question is, would you please comment on the toxicity of biotransformation products? And Jim, we'll start with you if you wanted to add anything more about biotransformation products or their anticipated toxicity.
Yeah, so this question has not really been addressed hardly at all. So a lot of times when people are making comparisons, they're making the comparisons with the conventional explosives, as you've heard at today's talks. However, to me an important issue is what is the toxicity of the daughter products that are being formed when these materials go out into the environment. So the answer is much more detailed in the case of DNAN. Clearly in the case of DNAN, the biotransformation products are either equally toxic to the parent compounds, in a few cases even more toxic, and in some cases less toxic, but not less toxic to the point where it's not toxic at all. It's less toxic, but it's still toxic. So we see a range of those things, and it sort of depends on what the target organism is, what the answer is. But we've done some testing of this with microbial assays, and also with zebrafish, and also with Daphnia.
So Jim, when in your discussion, you're referring to toxicity to organisms generally, correct?
Yes, and yeah, I gave you a range of answers that sort of a range of answers that takes into account different target organisms that we've been evaluating.
Okay, thank you.
If I could chime in, this is Mark. I think we can learn from other explosives when nitro groups are reduced and we get the monoamines. What the toxicity could be to higher level organisms such as vertebrates. It's not very straightforward. Typically, when these nitrates are reduced to these amino forms, they are typically less toxic because they're less water soluble. And solubility or kinetics are very important in toxicity to vertebrates. You know, how well are these things absorbed through the gastrointestinal tract is the question. And typically things that are very water soluble are much more easily absorbed and results in greater toxicity to things that aren't as very water soluble, generally. Often things that are not very water soluble are increased in terms of their fat solubility. It's not the case with energetics. Because of their high nitrogen content, they typically don't act like other organic molecules. They want to form crystals instead of being more fat soluble when they are reduced in their water solubility. So at least that's general trends that we've seen with other explosive compounds.
Thank you, Mark. And does that also address the first part of your answer? Does it address ATO?
Yeah, that's what I was thinking about, that and DNAN, because both of these, the nitrate groups are reduced to amines.
Thank you. Katerina, would you like
yeah,
to add
so,
anything? Go ahead, Jim.
Go. Yes.
Okay, I was just going to say that we studied this question in much greater detail with DNAN and as opposed to NTO, but we have done some studies comparing NTO with ATO. For most target organisms, so microorganisms is the one that we studied the most, we see that NTO is toxic and ATO is less cytotoxic. However, we also did a Daphnia test. So Daphnia as opposed to Cereodaphnia that was tested in Mark's presentation. In that one, we see that there is potentially a higher toxicity of ATO compared to NTO. However, we cannot send NTO to outside laboratories because of ITAR regulations. So all we can do is just compare our information with ATO with the literature, and unfortunately all we have in the literature is compare. So we tested Daphnia, and we have to compare it to Cereodaphnia in the literature. But based on that shortcoming, we see that ATO is more toxic for Daphnia.
Okay, and Katerina, would you like to add anything about the toxicity of the biotransformation products?
Yes, I would like to add something that is based on the little discussion we had just before the presentation. I was asking Mark about toxicity of these compounds to the not the products, or maybe products as well, to the microorganisms, because this has feedback on their transport behavior in soils, because we have some evidence, which is not very strong, but it's possible that we have lower rates of phototransfer, or not phototransformation, just regular transformation for solutions where we have high concentrations of these compounds than we have lower concentration. So it's possible there is a suppression of the transformation if you hit it with high concentration of NTO or DNN.
Okay. Thank you to all of our speakers for addressing that question. The next question for everyone is, considering all the properties of these insensitive munition compounds, their solubilities, toxicities, attenuation mechanisms, which compound do you expect would present the greatest environmental concern overall? And Katerina, we'll start with you.
Well, I don't have an answer to that. I have some thoughts that and some feedbacks from everybody. We talked about it before. Is that overall, the big concern with NTO, of course, that it is so very soluble, and so there is a fairly good chance that we'll have this entering into the groundwater or surface water as well. And so we would expect that it's very likely to reach the target organisms. So the question is it going to harm them when it does reach it? And it seems from what Mark has done is that the effect of NTO is very low on the, so that they have to see very high concentrations to see any effects, if any at all. So it appears that despite its very high solubility and mobility, probably the impact may be smaller than you would see for more traditional explosives.
Okay, thank you. Mark, do you have any thoughts?
Yeah, I would agree with Katerina that the NTO would probably be the biggest thing to keep your eye on. Again, because of the water solubility and the fact that making it to groundwater is not unlikely, and that also when you think about how we assess risk from compounds that do make it to groundwater or make it to water. Typically what the EPA does is they'll develop a reference dose, and you'll take toxicity data and you'll use uncertainty factors on those toxicity data to get to a chronic safe level exposure to the general population. A concentration you can be exposed to every day for over 30 years without risk of deleterious effects. And even though we don't really see effects until very, very high exposures, when you apply those factors in a risk assessment context, you get to fairly low concentrations. And so, the question in my mind is, how much can we use at a range until we get concentrations in water where, it could be an issue. That's why I think it's really important that we try to fill in as many toxicology data gaps as we possibly can, so regulatory agencies can feel more comfortable about what the probability for adverse effect would occur at certain dosage levels.
Okay, thank you, Mark. And Jim, do you agree NTO is the one to keep your eye on?
Yeah, I would agree with both Mark and Katerina. I think that the chance for NTO to get into the drinking water supply is much higher than the other compounds, and for the reasons that Mark stated, when you try to estimate, what a safe level would be with all those risk factors, then that it's going to bring it down to a concentration that's probably not unreasonably to find when it does get into groundwater. So for that reason, I think the NTO is the one that's going to get the most attention. However, there will be special situations. When you have DNAN as your predominant pollutant in soils with very low organic matter, then those are situations where you could have transport of DNAN, and so we have to pay attention to that also.
Okay, thank you. And thank you to all of our speakers for the presentations today and for this excellent discussion of all of the audience questions. Our next webinar in this series is on Thursday, June 16th, and will be the second half of the webinar series on the long clap for advanced geophysical classification investigations. The webinar will feature Dr. Jordan Adelson, the director of the Navy Laboratory Quality and Accreditation Office, and Dr. Ed Corll, who's the deputy director of the same office. Registration is open for this webinar, so please visit the webinar webpage to register for this and other webinars. Before we conclude, I would like to remind you that both the audio and a copy of the presentation of today's session will be archived on our webinar webpage in case you would like to refer to them in the future. We would appreciate it if you would please take a moment to complete the survey that will pop up on your screen at this time. This concludes today's webcast. Thank you.